The last week, briefly talk about the cardiac muscle. We will also mention a little bit when we talk about the heart. So the cardiac muscle, they combine properties of both skeletal and smooth muscle. First of all, they are striated muscle just like skeletal muscle, but they have branching processes. They do not like a skeletal muscle form a cylinder shape. Cardiac muscle have branching, and each branching with other, they lock each other. Adjacent cells with this branching, they are joined by a special structure called the intercalated disc. And on the intercalated disc, that's the fusing of two cell membrane of the cardiac muscle cells. There are gap junctions in between, just like single unit smooth muscle. The whole cardiac muscle, more or less like one unit, and that's also important. With gap junctions, with intercalated disc, all the cardiac muscle can be activated synchronously at the same time. And there's another difference between skeletal muscle and cardiac muscle. Although they are striated, they have organized Z-line, dark band, light band, dark band, light band. They also have quite well-organized sarcoplasmic reticulum. Okay, this is all similar. But the difference is on the cell membrane of cardiac muscle, they have a specific type voltage calcium channel called the L-type calcium channel. The depolarization of the heart muscle still depends on voltage-gated sodium channels. So when there's a depolarization comes on, the sodium channel open, sodium flux in, the membrane becomes depolarized, the muscle will start to contract, right? And so the calcium released from sarcoplasmic reticulum, and calcium will bind to thin filaments, troponin, and then remove the tropomyosin, and the cross bridge can form, so muscle can contract. But at the same time of the sodium influx induced the depolarization, the L-type calcium channel will also open. L means, you can say, long-lasting. You will have this long-lasting calcium channel opening. So calcium is also a cation, positive charged ion. So calcium flux in is also cause prolonged depolarization of the membrane. So with this long-lasting calcium channel opening in the cardiac muscle, the action potential will last much longer. So unlike the smooth muscle or any neuron, the action potential just goes up and down. If you compare the skeletal muscle, the excitation and contraction, excitation only lasts for a few milliseconds in skeletal muscle, but contraction can last for 100 milliseconds. And if you look at the cardiac muscle, the same thing, the contraction will last 200-300 milliseconds, the contraction. And also, you see, the action potential goes up by sodium channel opening, and then the depolarization will persist, forming a plateau. This is induced by long L-type calcium channel influx. And then this almost lasts as long as the contraction, and then will depolarize. What's the importance of this L-type calcium channel? As you know, if we stimulate the skeletal muscle before it's fully relaxed, you can stimulate further contraction with much more tension goes on, and then you can even get the unfused or fused tendinic contraction. But for cardiac muscle, tendinous or tendinic contraction is fatal. So the cardiac muscle, you contract, you need to relax, because the heart needs to eject blood and then relax to receive blood for the next ejection of blood. Unlike the skeletal muscle, the cardiac muscle cannot have a tendinous contraction. That will be fatal. So the prevention for that is you just prolong the depolarization, and that means you have all the way, it's a latent period for the next generation of action potential. So it will last as long as the contraction. When the contraction is fully relaxed, then you can have the next action potential comes in and the contraction of the cardiac muscle. We will emphasize that again when we talk about the cardiovascular physiology. Other than that, we can repeat the excitation, contraction, coupling in cardiac muscle. So the membrane is depolarized by sodium entry as the action potential begins. And this depolarization will open L-type calcium channel in the T-tubule, and small amount on the T-tubule first. Small amount of trigger calcium enter the cytosol, contributing to cell depolarization. And this trigger calcium binding to and opens ryanodine receptor calcium channel, opens the sarcoplasmic reticulum membrane. And calcium flows into the cytosol, raising the calcium concentration, and binding of calcium to troponin exposed cross-bridge binding site in thin filament. And then cross-bridge cycling causes force generation and sliding of thick and thin filaments. And then calcium ADPase pumps will remove calcium from the cell. First pump will return calcium back to the extracellular compartment, and the other calcium ATPase on the sarcoplasmic reticulum membrane will return calcium to the sarcoplasmic reticulum. And then the membrane is repolarized at the end of the action potential.